Hi students, in this video we are going to learn about the archegoniophore and archegonia structures of Marchantia. In the last video we have learnt about the anthridiophore and the anthridial structures. So now moving on to the archegoniophore. Like we have learnt in case of anthridiophore, archegoniophore is also a stalk-like structure which arises from the apical notch of the thallus. So it possesses a stalk and a disc at its terminal portion. This stalk is somewhat longer than the anthridiophore. Archegoniophores stalk is longer than the anthridiophore, and the disc is usually 8-lobed, the same way as in anthridiophore. So here the archegonias are arranged in acropetal succession. That was the same case in anthridiophore also. So here archegonia are also arranged acropetal succession, and there will be around 14 archegonia on each of the lobes of the disc. As you can see, this is a Marchantia thallus, and from this thallus arises a stalk, and this is the disc. From this disc will be having around 8 lobes, and in each of the lobes the archegonias are arranged acropetally. That is, what is acropetal succession? You know that, I hope. So they are arranged acropetally in these lobes, in this central disc. Okay, so in each lobe there will be around 14 archegonias, and what happens is, after the fertilization process, the stalk of the archegoniophore, this stalk elongates further, and what happens is the central portion of the disc, okay, the central portion of the disc, it grows more faster than the ventral surface. Okay, the central portion of this disc grows more faster than the ventral surface, and because of this change in growth, or because of this differential growth pattern on the two surfaces of this disc, what happens is these two surfaces will be showing difference in their growth pattern, and as a result of this, what happens is the archegonia bearing marginal regions of the disc, it will be pushed downwards. This marginal regions, it will be pushed downwards. Okay, this regions, the central regions or the dorsal regions, will be growing faster than the ventral surface. So what happens is more cells will be produced on this surface, and it causes the curvature, or it because of this change in growth pattern, it causes the disc to be pushed downwards and inward also. So in mature archegoniophores, archegonia lie on the ventral surface. This is pushed downwards, and the archegonias which were on this surface are now seen on the ventral surface of the disc, and they are inverted also. As you can see here, they are inverted also, the neck facing the downward position. The necks are directed downwards, and the process of this inversion, here the arrangement of archegonias are reversed from centripetal to centrifugal succession also. That is, the youngest archegonia, they now lie close to this central stalk, and the older ones are towards the peripheral region. And after this inversion, a one-celled thick plate is formed, which is known as perichaetium or involucre, on each of the sides of archegonial rows. So the archegoniums of the adjacent lobes are separated by these perichaetium. And in many of these species of Marchantia, long and stout finger-like projections are developed from the margins of the disc between the archegonial groups, and these finger-like projections are known as rays. These are the rays. Okay, these are the finger-like projections arising from the stalk disc is called as rays. And these rays, it gives the archegoniophore an umbrella-like appearance. And coming to the structure of archegonia are flask-shaped structures, and they are attached to the ventral or the swollen region by a small stalk. It has a venter region, swollen region, and an elongated neck. The swollen region is called venter and the elongated region is called neck. And venter is surrounded by a one-celled thick sterile layer, that is the jacket layer, and it contains inside this venter it contains one egg and a venter canal cells. This long neck region, it is enveloped by, or it is enclosed by the neck canal cells. These are the neck canal cells on which is filling the neck cavity. So the neck cavity is filled by the neck canal cells. It will be around 4 to 8 in number. And at the tip of this neck there will be 4 cover cells which will be arranged as a rosette. So this is about the archegonium. So archegonium is a flask-shaped structure. It has a venter and a neck region. Venter, the cells lining the venter region are called venter canal cells, and those lining the neck region are called neck canal cells. Inside the venter, it will be having one egg and a venter canal cell, and the neck cavity is filled by around 4 to 8 neck canal cell, and at the tip region it will be having 4 cover cells. So this is all about the structure of archegoniophore and archegonium. In the next class we'll discuss about the fertilization and the remaining portions. Okay, thank you.